So our next speaker is Dr. Benjamin Ryskeldi Falcon from the LMB. So Benjamin's had his research group at the LMB since 2019, and he's focusing on molecular mechanisms of protein assembly in neurodegenerative disease. And so Benjamin, please come up and present your structures of pathological protein filaments in dementia.
Thank you very much. So it's a pleasure to be here today. So yes, I have a group at the LMB up the road in Addenbrooke's. And I guess what I'm going to talk to you today about sort of boils down to neuropathology done at the atomic level using structural biology. So just to start, I mean, this will be obvious to many people here, but I just want to make the point that almost all cases of neurodegenerative disease has as their hallmarks the assembly of a limited number of specific proteins, most of which are shown here. And these assemblies form inclusions in neurons, in glial cells, and occasionally in the extracellular space in the brain. I think the important takeaway from this slide is that these proteins don't have any obvious common functionalities in health or obvious similarities in their structures in health. Rather, they're united by their assembly in disease. And the second point is that in the genes that encode each of these proteins, there are pathogenic autosomal dominantly inherited mutations that lead to assembly. So this really shows us that protein assembly plays a causal role in neurodegenerative disease. Now, despite knowing this, we still don't fully understand how these proteins assemble in the first place, why that happens, and how they then go on to damage the brain and lead to neurodegeneration. And for me, at least, part of the issue was the inability to study these assemblies at the molecular level using structural biology. So this had actually been a long time interest of Cambridge and of the LMB. So back in the early 1980s, Martin Roth at the Department of Psychiatry and Aaron Klug at the LMB set out to determine the structures of these protein assemblies. But it wasn't until 2017 that the labs of Michel Goede and Ryskeldi work that I was involved in as a postdoc, together with another postdoc, Anthony Fitzpatrick, where we were able to harness new advances in cryo-electron microscopy to then actually reveal the structures of these assemblies. And we did this for the protein tau in Alzheimer's disease. And you can see in this schematic, we extracted the filaments from post-mortem brain tissue from individuals who had died of Alzheimer's disease. And we were then able to use cryo-electron microscopy, so to freeze these filaments, bypassing the formation of crystal ice to form amorphous ice to preserve their structure in time and space, and then using helical processing algorithms, go from 2D projection images from the electron microscope to 3D structures. And so this, you can see here the structure of paired helical filaments of tau in Alzheimer's disease, these two C-shaped protofilaments. And what we found for tau was that they form amyloid filaments. And what we mean by an amyloid filament is a filament fold that's enriched in beta structure, so that when you stack molecules with identical structure on top of one another in a filament, you end up having these intermolecular beta sheets running along the filament axis. Often, although not always, we also see that the molecules fold back on themselves, and then you get the lateral stacking of beta sheets perpendicular to the helical axis. So following on from this work, the labs of Michel and Schurz have gone on to determine the structures of tau assemblies in almost all the neurodegenerative diseases that they're implicated in, as well as those of alpha-synuclein and also of beta amyloid. And in all cases, they find amyloid filaments with disease-specific folds. However, I'm going to talk to you about a different protein today that I was interested in when I set up the lab about 4 years ago, and this is TAR DNA binding protein 43. So in 2006, this was shown to be the assembled protein hallmark of almost all cases of amyotrophic lateral sclerosis and about half of cases of frontotemporal lobar degeneration, which of course lead to frontotemporal dementia. They also underlie a quite recently described disease entity, limbic predominant age-related TDP43 encephalopathy, or LATE, which is aptly named because this is thought to be a clinical mimic of Alzheimer's disease, late-onset Alzheimer's disease, where instead of tau and beta amyloid driving the disease, we have TDP43 assembly. And there's estimates of around 15 to 25% of cases of Alzheimer's disease with an onset over 80 years old may be attributable to this condition. We also see TDP43 assembly as a co-pathology in many other neurodegenerative diseases. Probably over half of Alzheimer's disease cases have some limbic TDP43 assembly as well. And it's been shown that when you have this assembly, that leads to worse disease outcomes. Now, we got interested in this because unlike tau and alpha-synuclein and beta-amyloid, it was thought that TDP43 did not form amyloid filaments. So 4 years ago, here's what we knew about assembled TDP43. We knew, like many of the other proteins in neurodegenerative disease, that it accrues abnormal post-translational modifications. And the two most striking of these are the truncation of the molecule from the N-terminus. So we lose the RNA recognition motifs, the nuclear localization signal, and the Dix domain. So these are key for the functions of TDP43 as an RNA and DNA binding protein. And we keep fragments that contain some of the RNA recognition motifs and all of this low complexity domain. And the low complexity domain just means we have an uneven distribution of amino acids. And in the case of TDP43, we have an enrichment in glycines, glutamines, and asparagine residues. The second abnormal modification was phosphorylation at the extreme C-terminus. So there are 6 disease-specific phosphorylation sites confirmed to date, and they all cluster here in the second half of the low complexity domain. So back in 2008, just after the discovery of TDP43 in disease, multiple groups had used thin section fixed EM to look at the inclusions that TDP43 forms within neurons and glial cells. You can see here at low magnification one of these spherical inclusions that was in a neuron. You can see it's a meshwork of filaments, and if you use antibodies against TDP43 conjugated to gold molecules, you can see that they are formed of TDP43. So far, so similar, we have filaments of TDP43. However, there are a couple of observations that were a bit odd. And the first one was that these filaments do not bind compounds that typically bind to amyloid. So we're thinking about Congo red, thioflavin T, and their derivatives like the PET ligands used for tau and beta amyloid today. And the second puzzle was that it was very difficult to isolate this assembled form of TDP43 in disease away from the physiological form of TDP43. And that's because TDP43 also assembles in health. The Dix domain is a ubiquitin-like fold that can form filaments by head to tail stacking. And there's some evidence that filament formation or oligomer formation through the low complexity domain is also important to help TDP43 bind to long RNAs. So the first thing we wanted to do is to develop a protocol that would allow us to isolate the disease-specific TDP43 assemblies to a purity and yield where we could do structural biology. And so this was the project of the first postdoc to join my group, Diana Arseni, a fantastic postdoc who did most of the work I'll show you from now on. And essentially, she took the protocols that work well for tau and alpha-synuclein, but did not work for TDP43, which are based on differential centrifugation, the use of detergents to solubilize some of the assemblies from the brain. And she tweaked this until she was able to show biochemically that she could leave behind the physiological form, the full-length form of TDP43, shown here in this soluble fraction, and enrich in an insoluble fraction only the disease-associated form, which we see is phosphorylated with antibodies against phosphorylated TDP43 and contains this C-terminal fragments of the protein. So when she then put that on an electron microscopy grid and used gold-labeled antibodies against TDP43, she only saw filaments of TDP43, no other globular assemblies that had been seen with previous extraction methods. And this then enabled her to make cryo-EM grids and to perform cryo-electron microscopy on these preparations. So the first thing we do when we're looking at the cryo-EM data is we start to average together the 2D projections to get an idea of the heterogeneity within the structures. And so this is done in an unbiased manner, so it will class similar structures together. And for TDP43, we only got a single 2D class average. You can see it's filamentous from left to right, and there's some indication of a helical twist to this filament. But of course, we can progress from there into a 3-dimensional structure, which looks like this. So we have these rungs along the TDP43 filament. That's individual TDP43 molecules. So that looks like an amyloid filament stacking up on top of one another. And if we then have a look at the cross-section of this filament, we can then see how the TDP43 molecule winds its way throughout this fold. And at the resolution we are at, about 2.5 angstroms, we can then sequence the protein from these structures based on the amino acid side chain densities that we can see. So before I go into the structure, I just like to say that this was extracted from the brains of individuals who had ALS and FTLD. I'll talk a bit more about that type of disease later. But we were keen to look at multiple individuals. So initially we looked at 2 individuals with this disease and found the same fold. And then we looked at the motor cortex associated with the presentation of ALS, and also the prefrontal cortex associated with the FTLD. And again, we saw an identical fold. This is then the atomic structure of TDP43 built in to that cryo-EM reconstruction. This shows us that it is the first half of the low complexity domain that forms this fold. That makes a lot of sense because it can incorporate all of the disease-associated truncated forms of TDP43, and it can incorporate the phosphorylation sites. We'd expect them not to be in the core because then there wouldn't be access for kinases to carry out this phosphorylation. And you can see it's indeed low complexity, quarter glycines, third neutral polar residues, third hydrophobic, only 1 charged residue in arginine down here on the surface of the fold. And actually we can see that most of the hydrophobic residues are in the center, so they're buried in these 2 hydrophobic pockets. And then we have a glycine-rich region at the end terminus and the glutamine and asparagine-rich region, which form these spiral arms wrapping over this, what we call double spiral fold. So one thing that came out of this is that actually the surfaces of these filaments are very distinct, and this from those of tau, for example, or alpha-synuclein. And this might explain why the compounds that typically bind to amyloid filaments do not bind to these amyloid or amyloid-like filaments of TDP43. So for tau, you can see a good distribution of charged residues on the surface. For TDP, we only have a single charged residue. And it's been shown by Michel Goede and Scherz that if you look at PET ligands that see these tau assemblies, they like to bind within these grooves on the filament surface where we have a beta sheet and we have large charged side chains. And these simply do not exist on these TDP43 filaments. Having said that, that doesn't mean that there's nothing that will bind these filaments. We do see in the raw structures additional molecules pointed to with these arrows that are not formed of the protein itself. And I just focus on this one with the 2 yellow arrows. So this is a very flat surface formed mainly of glycine residues, and we see planar molecules that line up with very high occupancy against this surface. So we think that we can use these structures now to have a new generation of imaging tools that will see these filaments. So having looked at ALS and FTLD, we're interested in other diseases where TDP43 assembles. And so we focused on FTLD, frontotemporal lobar degeneration. And there are at least 4 types of FTLD characterized by TDP43 assembly. They're labeled A to D. There's possibly additional types, a type E, a type U. But focusing on these types, they're distinguished by the distribution of the TDP43 inclusions in the brain. So in type A, you have cytoplasmic and nuclear inclusions only in layer 2, or almost only in layer 2. In type B, C, and D, you have distributed throughout the neocortex, but you have only cytoplasmic in type B, neuritic, definitely axonal, potentially dendritic, and only nuclear in type D. We also see different genetic variation that can predispose to these different types of FTLD. Granulin mutations are unique to type A FTLD, whereas C9orf hexanucleotide repeat expansions can lead to both type A and B. There's no known genetic variation for type C, and then VCP mutations cause type D FTLD TDP. And so these different neuropathological presentations of TDP43 assembly then lead to different but overlapping clinical presentations of FTD. Just want to point out that in type B, that's uniquely associated with ALS. So these were the cases I showed you earlier that we focused on. And in type D, we have the multisystem proteinopathy that can present as a myopathy or a motor neuron disease. So yes, we focused on this type B, and we now went on and we're interested to look at type A. And the hypothesis was that perhaps different structures of TDP43 could account for the differences in neuropathology and then the ensuing clinical symptoms. So I'll skip through the extraction of the filaments. We did this in the same way and from the prefrontal cortex as well, similar to the type B cases. And I'll go straight to the cryo-EM structures. And we looked at 5 individuals this time. And you can see that we have an identical structure for all 5 individuals. And we looked at 2 individuals with granulin mutations, 1 sporadic, so had no known genetic mutations known to cause or to increase the risk of neurodegenerative disease, and then 2 individuals who had repeat expansions in C9 orth, and we have the same structure in all of them. This is what it looks like. So again, it's pretty much the identical part of TDP43 that forms the filaments in type B FTLD, that first half of the low complexity domain. Again, it's burying the white, shown in white here, hydrophobic residues, with the glycine and the glutamine and asparagine rich regions covering these. But you'll be able to see it's a completely different fold, which I'll come on to later on. Just one interesting thing about these filaments was when we started to process the data a bit further, we could actually find there was local alternative conformations of this fold. So in cyan, I show you the main conformation we saw in the previous slide, but we could actually find rare conformations where we had different turns and loops, either at the end terminus of the fold or at what we called the turns linking the layers. So we think of this as a 5-layer fold from top to bottom, and we also have an alternative conformation here. And the benefit of cryo-EM is we can actually go back and see where in the raw data did these structures come from. And when we did that, to our surprise, we found that actually we have 2 filaments here. These alternative conformations could coexist within single filaments. And that's a sort of artistic representation of what that would look like, stacking up 2 TDP43 molecules with each of the different local conformations. So this shows us that these filaments are not completely fixed, rigid structures, but they can change along their lengths. And this is important if we think about designing tools that will bind to these filaments for diagnosis or therapies. We don't want to target these regions. The filament may just change its structure. Rather, we should go after these fixed and rigid regions. So this is then a comparison of the 2 folds that I've shown you in type A FTLD and in ALS and type B FTLD. And you can see they're formed by almost the same sequence, but have very different folds. And they're different in their secondary structure motifs, so regions that form loops and turns in one fold may form beta strands in another, but also in the trajectories that these different turns take. And this is really bringing us to a broader notion in neurodegenerative disease, as was shown for tau and alpha-synuclein, that we can characterize different neurodegenerative diseases based on distinct amyloid folds of specific proteins. So I show you here the work that's been done for tau and essentially all the tauopathies, for alpha-synuclein, and we can now add TDP43 here as well. And so this has implications for designing tools that may see all of these assemblies based on their commonalities, or may be able to pick out distinct assemblies for disease-specific applications. So I'll stop there and just thank the many people involved in this work, especially Diana Arseni, who I mentioned earlier, who drove the cryo-EM forward in the lab, and the PhD student Heidi Chen, who carried out the mass spectrometry of these assemblies to confirm the protein identity, and something I didn't talk about today, but some of the new post-translational modifications that these structures throw up as well. Also, thanks to the neuropathologist collaborators who provide us with the characterized brain tissues, and to the patients and their families who donate this tissue, and finally to my funding sources. Thank you very much.